great. Thank you.
All right, very good. Okay, well, thank you. Thank you for inviting me, and I am sorry I couldn't be there. I really did want to be there. I am not a bench scientist. It's been about 29 years since I did that. And full disclosure, I was not very good at it. So I moved into clinical research, and that's where I spent my career. I was at 2 or 3 academic institutions until about three or 4 years ago when I set that down and joined the FDA. So I'm a transplant hepatologist, but my area of research for the last 15 to 18 years has been DILI, and particularly how to diagnose DILI from a clinical point of view. So that's why I'm very interested in this workshop. I think I'm getting more out of it than I maybe can give you, but nevertheless, they asked me to give this perspective. So these are my disclaimers. This is not an official FDA point of view, anything that I say here. So I talked to Mark Avigan, the past couple of days, and he gave a talk, very nice talk. He's an expert in DILI pathophysiology. So I didn't want to overlap much with him. And then Dr. Cohen gave a very nice clinical view of DILI. So that left me to just sort of give you a fairly high overview of who we are, introduce the DILI team, because the FDA didn't have a DILI team until about 3 years ago on the pre-market side. And then talk about basically why we think non-clinical information, including CIVM, is very important to us, and how we use non-clinical information right now in our workflow. So first of all, here's the DILI team workspace. We reside in the Division of Hepatology and Nutrition. The 2 dark blue lines are our 2 inboxes. They constitute probably 90% of our workload. NDA and BLA consultation and IND work. But up in this upper box is also tests and devices from ODS and CDRH. Now, we are consultants. They have the final say on the decision, but they do consult us fairly routinely for devices and tests that relate to DILI. I want to point out a couple other people. We do lean on non-clinical people quite a bit. We have a fairly small team, as you'll see. Minjin Chen at the National Center for Toxicologic Research in Little Rock, Arkansas. I'll come back to some of his work. We also have Fred Mullen, who is a true blue toxicologist, and Yuqing Yang, who is an expert in quantitative systems technology. So while we're a small team, we have built bridges to other people within the agency at large to help us out with the work that we do. And I'll say something special about the closest relationship we have, which is with OSC, and that was Mark Avigan, who spoke yesterday. So since the DILI team has stood up, we have tried to forge a much closer relationship with OSC. We are on the pre-market side. They are on the post-market side. And as you can imagine, it's very important for us to stay in communication. We meet every other week. For example, we may have a drug that we're going to put over the line and say it's going to be approved, but it has a DILI risk liability. We obviously get OSC involved early on to let them know it's coming and how they're going to surveil and even help, maybe even mitigate that risk. On the other hand, if they see a signal on the post side, and we're dealing with 2 or 3 drugs in the same class, well, obviously we want to know about that too. So this is a very important relationship, and it makes a lot of sense. So I'm very proud of the fact that we shored this relationship up since the DILI team stood up. But we are small, at least right now. There are only 4 members on the DILI team at its core. I bolded the last person, who basically looks at all their non-clinical data, and she would be looking at any CIVM data that would be coming down the pipe, and hopefully we will see it coming to us. So then on to sort of the major points, of which aren't that many, just a couple that I want to make. You've seen this slide in various iterations and forms. My point here by showing it, though, is I think most of the talks have been dealing with this step, how to move from It's, in other words, I think I heard it said, kill the drugs that you need to right away and move from non-clinical into clinical into human beings. And that's all fine and good, but I have to, and we do get consulted at that level as well. But really, the lion's share of what we do is down here in the clinical trial space, INDs, NDAs, post-market, when we have a signal that we're having to deal with. My point is that from our perspective, we reach back, no matter where we are in the process, we reach back to that non-clinical data. And I'll tell you why in a bit. You'll see why that's important to us, particularly when we're still in the pre-market space, the approval process, which is a lot of our work. What it comes down to is we have to make the diagnosis of DILI in individual patients a lot of times. And that can be critical. I like to say it's a sort of a tight margin or a low threshold. It doesn't take many cases of DILI, severe DILI, to jeopardize the whole program for approval. So it's important to talk about is how we make that diagnosis as best we can and as accurately as we can under the circumstances we have. And we will take any data that we can, including non-clinical. So you saw this plot yesterday, but let me just read what's in our guidance regarding Hy's Law cases. This is, first of all, the plot is the standard screening plot for hepatocellular DILI. It's been used since the 2000s. It's sort of been the anchor of DILI assessment at the FDA since then. But basically, finding one Hy's Law case in the clinical trial database is worrisome. Finding two is considered highly predictive that the drug has the potential to cause severe, i.e. fatal, DILI when given to a larger population. Short translation, all it takes is one or two cases and we got a problem. This is a large trial you're seeing here. A lot of time, effort, and money has been placed. But if even two of those cases, we come in and we assess two of these cases as attributable to the drug, then we all have a problem. So that is why it's really critical to talk about how best we can make that diagnosis as accurately as we can, because it is very, very important and can upend the whole boat if we call those DILI cases. So how do we do that? Well, that's the added challenge. We don't have a blood test. We don't have a histology biopsy that will say this is DILI and this is not. People are working on it, which maybe some in the room are even working on that, but we don't have it right now. So what we use is what's called just subjective expert opinion. And it ends up being, the output is a causality score. This is what the FDA now uses. This is what is being used in industry. As far as I can tell, this is the outputs they give us. It's based on the Drug-Induced Liver Injury Network, which is a UO1 NIH-funded study. It's been going on about 16, 17 years. I've had actually the privilege to be a part of that for the last 15 years, and I'm actually co-chair of the causality committee. So what our outputs are, and what we do at the FDA as well, is we give a probability, a causality score. But I wanted to point out that these scores, they're not just subjective categories, they have a semi-quantitative likelihood percentage. That's the sort of the context of how we look at it. We try and figure out what's the percent likelihood, like you're betting, and you want to put a percentage down. We're going to use this, and then we get our causality score. The other thing I want to point out is that this language over here, it's really sort of straight out of an episode of Law and Order, right? It's beyond a reasonable doubt, clear and convincing, preponderance of evidence. I emphasize this because it is a body of evidence issue. And what does that mean? Well, that means that we will take the non-clinical evidence. We will look at it. Because right now, it's the best we have for a pre-event probability, which I'm going to cover now. So if you think of a DILI diagnosis as a table, it stands on four legs. The body of evidence can be broken down into four major legs. One, timing, pretty straightforward. Patient was on the drug for a certain amount of time, had an injury, it's consistent or not. Pattern of injury, is it cholestatic? Is it hepatocellular? Was there jaundice? And exclusion of other causes. We don't want to call hepatitis E a DILI, although they can look very similar. The last one is pre-event risk of DILI. I use that term because it's a little less intimidating than pretest probability, but that's all this is. And I know Dr. Cohen brought this up. I was pleased to see him put that forward, and I think Dr. Strauss also brought it up. See, in clinical practice, we have all four legs because drugs have been out on the market for a little while. We can look them up on PubMed. We can go to LiverTalks or other places and get a sense of what the risk for that particular drug is, even before the event happened. When I came to the FDA after doing this in clinical practice, I realized that 3 of the legs were intact on the pre-market setting. And in fact, they were even better. A lot of times we have better timing data. We have better pattern of injury data because they're being followed in a study. But what we lost was that pre-event risk, that pretest probability, because they're new drugs. Now, besides the fact that this table looks quite unstable, you would not want to lean on it. But I would argue that of all the legs to lose, that was one of the most important. Why do I say that? Well, that leg informs how we look at the data from all the other 3 legs in a major way. And that's what pretest probability does for you. Let me show you this concept in a different way. So here is a post-event data. Again, the DILIs, something's happened, a liver injury's occurred. These are the 3 legs I mentioned: timing, pattern, exclusion. Now, if I don't get any pre-event likelihood, which is where we exist more or less in the pre-market setting, the way this works out, you don't know if the positive predictive value is high or low. On the same side, you don't know if the negative predictive value is high or low. That's why you need the pre-event likelihood. Okay? So that is why we are interested in what's going on in all this non-clinical stuff. Let me give you a clinical example to show you this, how this works, and I think you'll see. So let's just say you had a DILI injury, a liver injury. The timing, the pattern, exclusion all point toward DILI. They're all positive. They're saying this looks like DILI to us. Now, let's say the drug is one with a relative high pretest probability, amoxicillin clavulanate, arguably the most common drug to cause DILI in the United States. Because of its high pretest probability, I will look at this data in a very different way, right? I will say, I'm probably up in this zone with a high positive predictive value, and I am likely to say it's highly likely, at least highly likely or probable, that this is augmentin DILI. And I'll probably be right. Now contrast that with amlodipine, very commonly used antihypertensive, but it doesn't cause DILI. It's very, very rare. So if that's the drug, I'm going to look at this data in a very different way. I'm going to know in the back of my mind that I have a low positive predictive value. In other words, they're positive, but are they true positives? Low chance. And I will come in at possible or unlikely. I'm going to say we got to look for something else, or we will just leave it as unknown, because it's unlikely these positive values are true. So that's how it is in clinical practice, but in pre-market setting, this is where we are. We're back to unclear. But what do we have? What makes sense? We'd lean on the non-clinical data, and this is what I meant by we have a injury showing up in trials, even post-market. To help us out, we will reach back, and if the CIVM data can help out, all the better, to help us get past and help us with this predicting where we are in this positive and negative predictive value scale. So I just wanted to emphasize that, and that's basically why I'm interested in this workshop so much. So moving on, what do we do actually in our consults now with the data we have? So our consults are fairly lengthy, and we've dedicated a section 3 to just non-clinical data. So we look at the non-clinical portfolio in depth. We look at everything we can to find anything about a DILI risk. It looks like this in our consult notes. We cover in vitro data, the animal toxicology data, we give a summary, and then we have a summary table that kind of lists the things that we think are most important. A lot of these things are familiar to you and were mentioned yesterday. So then let me just give you an example of what we do, or excuse me, an example of a model that I kind of like because it took non-clinical data and went all the way to the outcome that we're really interested in, which is what's happening in the label or post-market. This is post-market sort of outcome based on preclinical, non-clinical, at least 2 non-clinical data points. This was done by Minjin Chen at the National Center for Toxicologic Research. Maybe a lot of you know about this. Study was published in 2016. He basically had hypothesis driven that these amongst some others would be predictive. And then he took, I think, roughly 200 labels, and he categorized them as most concerning, drug was withdrawn, box warning, or warnings and precaution for severe liver injury. No concern was no mention of liver injury risk at all, and anything else was in between or less concern. And he ran it through this model and came up with a DILI score just based on three variables, 2 of which are these preclinical, non-clinical data points. And with that, he was able to produce this DILI score performance, where the DILI score is on the y-axis, the categories are on the x-axis. And as you can see, it discerns fairly well. It's not perfect, there's a lot of overlap, but at least it has the ability to have some idea of some cutoffs. For example, 6 and above, he considers high risk, 3 to 6 moderate, and less than 3 low. So this is not definitive by any means. Obviously, there's a lot of incorrect answers that you may pull from this DILI score. But on the other hand, it is some data that has significance and that we can use, or we will use occasionally in our consults. And this is an example of such a use. And I think this was mentioned before as one of the sort of uses for CIVM is when you're comparing drugs. This is an example. Obviously the drug names, and I fiddle with the numbers to de-identify it. But this is the kind of output we will get from Minjin Chen's group. The DILI scores along the bottom. Remember, greater than 6 is high risk. 3 to 6 is moderate. And it's based on these data up here, lipophysis, dose, etc., and which drug, where it is in development. So we use this data, for example, if, for example, there are 2 drugs out on the market already, and a third's coming down the line, how does it compare, for example, to the drugs and what they're seeing post-market? So this has been fairly valuable to us to at least give something in that pretest probability space to hang our hat on, or at least see if it coincides and lines up with what we're seeing clinically. So, as I said from the beginning, I was able to keep it fairly high level and keep my talk fairly short. The summary, a bottom line for us is that from our point of view, from the clinical DILI team's point of view, CIVM data could directly or indirectly inform clinical diagnoses of DILI pre and post-market. That's really what we're interested in, if it can do that. And we are supportive of its development. So if you all can put some of these tests over the line and validate it, et cetera, right context of use, you will have a receptive ear, at least from the DILI team, in terms of how we might use it and incorporate it. And with that, I will end. If there's any questions, I'm happy to do online, but I don't know if you guys are moving on or not. Thanks. Hello?
Yes, I think we've got time for one or two questions if there are in the audience.
Hi, Skip. This is David. Thanks so much for your talk. It was really great, and yeah, I know you sent me an email with some questions. Last night. I look forward to following up with you on those. I think
Great.
there really are a lot of parallels to the situation, and as I alluded to yesterday, in the cardiac QT torsade de pointes space, and I'll note it was interesting that you went into the pretest probability and how you think about applying, a diagnostic test. And yeah, I had referenced in my introductory talk yesterday, there were two articles, actually they were both in Clinical Pharmacology and Therapeutics in the same issue. The first one was a perspective from a group from the pharmaceutical industry. It included, I don't know, the 8 people that were sort of representing pharma and the FPA in Europe, and I think and the Japanese industry group in addition, that were on the ICH group and others. And in that paper, they presented really applying concepts of pretest probability and how to combine multiple tests together. It's using basic like Bayesian statistics and likelihood ratios. And then in our FDA paper, we actually, initially I was invited to write an editorial, but it turned into to be so long. It was a review article, and then there was another editorial from the editor of the journal on it, but we kind of reanalyzed their assessment in our article. And it has these concepts. And it's something that I think a lot of, it's hard to grasp but there's actually like quantitative ways to combine multiple tests together. And like, you don't have to have one perfect test. If you have like 2 tests, you suddenly you can have, you have, and I think you're kind of getting at that there. And yeah, it might be things for the community to think about further here going forward.
Yeah, I wanted to point out, it wasn't the purview of my talk, but as far as I'm concerned, that principle of pretest probability will apply, I believe, depending on where you put your CVM in the process line. In other words, if you do it before the animal studies, like you're just screening a bunch of drugs, or you do it after the animal studies, where some drugs have already been screened out, that will change your pretest probability. That will change your positive and negative predictive value. And I think I didn't see that mentioned, but I think that is an important concept, right? If you screened out the drugs that were in animals had injury, and you're left with a lot of that seem safer, and then you do your CVM, where your negative predictive value is going to be higher. So it makes a difference. On the other hand, if you test the ones that were toxic in animals, your positive predictive value is going to be higher. So it's all about enrichment and where you put it in the process of your non-clinical studies. So I did want to point that out. Thanks.
Yeah, I wanted to ask a question that goes back to the consult form that you have, and you had these farm talks, sort of like non-clinical data that you also include in there. And in the discussions yesterday, the presentations, it was mentioned that there are a lot of methods already available, in vitro methods, to look at reactive oxygen species and BSEP inhibition, mitochondrial function, various types of things. So in your opinion, do you think that if there were to be in vitro methods that assess some parameters of immune function in the liver, how would that impact your pretest probability kind of assessment? Might it do that, or might it not have any impact? Because I think that that's what the direction that we're trying to go in, is to try and identify specific assays that are going to be able to provide more information to you in order to make your decision as in the consult. And so I was interested in really being able to identify how to best design the assays to get the type of involvement of the immune system kind of parameters identified in order to impact that decision even slightly.
So first of all, yes, as I said, we would be open to hearing about that. And your operative word might it. Yes, it might. I would hope it would. Again, it's going to come down to the hard work you all have to do, looking at validity, looking at reliability, which was mentioned in the talk just before me. Are you doing the appropriate checks on your system consistently so that you're going to get the reliable test each time? All those things have to line up, but absolutely. Because right now, I have to be honest with you, when we get a biologic, that table is quite empty, right? As you might imagine. Those apply to small molecules. So Edwidge, my non-clinical analyst, she doesn't, it's an easier assignment for her because there's often not a lot in there. So we are open to that. But again, the proof will be in the details and whether you all can do that and bring it across the line for us.
Thank you very much.
Sure.